In November 2025, as the first wave of large-scale human trials for rapamycin and senolytics begins to report interim data, and as the first gene therapy candidates for partial epigenetic reprogramming edge toward the clinic, the biology of aging has reached an inflection point. What once seemed a scattershot collection of promising pathways has resolved, under the pressure of rigorous human evidence, into 5 interconnected mechanisms that repeatedly demonstrate measurable effects on health span and, in some cases, lifespan itself. The first of these is the inhibition of mTOR, the nutrient-sensing kinase that coordinates growth with resource availability. When chronically active, mTOR drives cells into a state of hypertrophic, inflammatory senescence. When appropriately suppressed, it restores autophagy, reduces systemic inflammation, and improves immune surveillance. The most studied intervention remains low-dose rapamycin. A series of 2024-2025 human studies, most notably the Pearl trial, published April 2025 in Aging, and a proof-of-concept pilot from the University of Texas Health Science Center, GeroScience, September 2025, have now shown that intermittent dosing, 5 to 6 means once weekly, is safe for up to a year in adults over 65, producing sustained reductions in inflammatory markers of 30 to 40%, gains in lean muscle mass, and subjective improvements in energy and well-being. A separate placebo-controlled trial of short-term rapamycin in older men, Moody et al. GeroScience 225 documented improved cardiac and endothelial function within weeks. Perhaps most intriguingly, when rapamycin is combined with the MEK inhibitor trametinib, an FDA-approved cancer drug, the two compounds act additively in mice, extending median lifespan by more than 30%, while preserving heart function and reducing tumor burden, Giccioni et al., Nature Aging, May 2025. The translational implications are clear. Dual pathway inhibition may soon enter human testing. The second pathway is AMPK activation, the counterbalance to mTOR and the cell's primary energy sensor. Metformin, already taken by tens of millions for type 2 diabetes, remains the most accessible pharmacological lever. A 2025 meta-analysis DC of 19 randomized trials, the Lancet Healthy Longevity, confirmed an average 8 to 12% improvement in insulin sensitivity and a reduction in epigenetic age scores equivalent to several biological years. The long-awaited Targeting Aging with Metformin TAME trial, now in its 6th year, has released preliminary data suggesting that metformin delays the onset of multimorbidity in non-diabetic adults aged 65 to 79. While metformin's effects on frank lifespan remain modest in healthy mice, its ability to lower HbA1c and thereby reduce glycative damage appears to be one of the few interventions with consistent human evidence across metabolic, cardiovascular, and neurodegenerative endpoints. Third are the sirtuins, a family of NAD+-dependent deacylases that maintain genomic stability and mitochondrial integrity. Sustained caloric restriction remains the gold standard for activation, but validated NAD+ precursors now offer a practical alternative. A 12-week Johns Hopkins trial completed in 2025 demonstrated that a combination approach, modest caloric restriction plus an NAD+ precursor, reduced participants' Horvath clock epigenetic age by an average of 3.1 years. More importantly, STI, a multi-center Alzheimer's trial using a cocktail that included the NAD+ precursor nicotinamide riboside, NR, reported reversal of certain neurodegenerative markers and improved mitochondrial function in early-stage patients. 2025 interim data. Direct sirtuin activators remain elusive, but the NAD+ sirtuin axis is now one of the best substantiated longevity pathways in human beings. Fourth is the clearance of senescent cells. The intermittent combination of dasatinib, a tyrosine kinase inhibitor, and quercetin, a plant flavanol, has progressed from first-in-human studies in 2019 to multiple Phase 2 and Phase 3 trials by 2025. Interim results from osteoarthritis cohorts show a 67% reduction in senescent cell burden in synovial tissue, accompanied by significant gains in physical function. A pilot trial in mild cognitive impairment, eBiomedicine, February 2025, reported feasibility, safety, and tentative improvements in MoCA scores, correlated with reductions in circulating TNAD. Long-term murine data continue to accumulate. Chronic intermittent depletion clears senescent cells from naturally aged mice without detectable off-target toxicity, improving vascular elasticity and renal function. Finally, and most provocatively, partial epigenetic reprogramming. The landmark Yang et al. Study in Nature Aging, Advance Online, November 18, 2025, demonstrated that a single week of controlled OSKM expression in naturally aged mice extended median lifespan by 6.4%, reverse epigenetic age by roughly 3 years, as measured by multiple mammalian clocks, and restored organ function across kidney, liver, skin, and spleen without inducing tumors. This is the first unambiguous demonstration that transient Yamanaka factor expression can extend lifespan in physiologically aged mammals, not merely in progeroid models. Human translation is accelerating. Youth Biotherapeutics received FDA feedback in September 2025 on its brain-directed OSKM gene therapy, YB-002, for Alzheimer's, with first-in-human studies projected within 3 years. Parallel chemical reprogramming approaches, small molecule cocktails that achieve similar epigenetic resetting without viral vectors, are in late preclinical development. These 5 pathways are not theoretical curiosities. They are increasingly interlocking. Rapamycin enhances autophagy, which in turn facilitates senescent cell clearance. AMPK activation raises NAD+ levels, feeding the sirtuins. Partial reprogramming appears to reset the epigenetic drift that gradually silences all of the above. The most sophisticated programs emerging from the Buck Institute, Altos Labs, and a handful of academic clinical consortia no longer target one pathway in isolation. They modulate all 5, often with surprisingly modest interventions. The evidence is no longer speculative. It is merely unevenly distributed. What remains is the hard, methodical work of translating these insights into safe, accessible, and equitable therapies. For the first time in the history of medicine, the biological clock is no longer running in only one direction.